No.
Oh, Marina's here. Carolina's here. I had no idea. I'm just gonna go be a Katie and take my car because it hasn't been running for a while. Yeah, that's fine.
Okay, can you hear me?
Yeah,
All right, sorry, there's a whole bunch of distractions going on here right now.
that's okay.
But me. Okay, so this is the, can you see the metabolism PowerPoints?
No, it's not showing it.
Okay, should be now.
Yeah, there it is.
Awesome. Any questions about anything?
No, I don't think so.
Okay. Let me just erase this. Let's go. Okay, so we left off talking about spontaneous versus non-spontaneous reactions. And just remember that, so for the test, I will give you guys the formula. You just need to know the what each of the terms mean. So delta G is Gibbs free energy. Delta H is the enthalpy, which is in the energy in the system. And we're talking about chemical reactions. This is energy in the chemical bonds. That's really the only energy available. And then T, that's temperature. And then delta S is entropy, which is disorder in the system. Okay, so remember the formula is delta G equals delta H equals minus T delta S minus. And so we, in order to get a non-spontaneous reaction to become spontaneous, like, you know, burning gasoline, for example, this value has to be negative. And so we can make it negative by decreasing the energy in the bonds, increasing the temperature, which, you know, makes sense. If you want to light some gasoline on fire, then you can increase the temperature, or you can increase disorder. And these are connected because if we increase temperature, we have to increase disorder because of the kinetic energy of the molecules. So we talked about in biological systems, we can't really use this because we can't change temperature, and we can't increase disorder in living systems because that will cause them to die. So this is really the only thing that we can change in living systems inside the body. So a lot of experiments and stuff are done outside the body so that we can manipulate the temperature and the disorder in the system to get the reactions to go. And later on, we're going to do that when we copy DNA in a test tube. Since it's not in the body, we can use temperature to get the chemical bonds to break so that we can sort of open the book of DNA. Remember, it's a double helix, and then we were able to read it. So because we're not chemists, we don't use exothermic and endothermic. We use this word gonic, which refers to energy. So exergonic reactions, exo means out, so it gives off energy. All right, and so you could think of this as digestion or cellular respiration. This is when the reactants have more energy than the products. They have less energy. So this is the energy in the molecules, right? So the reactants have more energy than the products. And remember that first law of thermodynamics says we can't create or destroy energy. So if the reactants have more energy and the products have less energy, then the difference of that energy has to go somewhere. And so its output, it can be output as heat, which is a form of energy. It can be output as another form of potential energy. Potential energy. This thing is terrible for writing. Which is chemical energy. And our chemical energy is ATP. So generally, when we do this sort of reaction for respiration, we're going to output it. The amount of energy output is going to be in the form of ATP. And you're always going to lose some energy as heat. And remember, heat energy is disordered energy because it's just randomly flying around. So if you just think about heat, it would just escape into the universe and just randomly go wherever. So it's not really a very useful form of energy. And it falls into this delta S category. So what you should take away from this is that the products have less energy than the reactants. And so you can sort of think of a rock would roll downhill on its own. So this is a spontaneous reaction, and that means the delta G has to be negative, because remember, this has to be negative to be a spontaneous reaction. If it's positive, it's not spontaneous. So energetically, things go downhill. It's sort of the natural way of like rocks or water would go downhill. Okay, so here's an example of this. So we take in energy from the food we eat. So we're going to just use glucose as an example. This is glucose. And we breathe in oxygen. And everybody learned this in probably 6th or 7th grade science. And we give off carbon dioxide and we produce water as a byproduct. And we're going to cover how all this works in chapter 9 in detail. But for this particular lecture, the energy in these are the reactants, is greater than the energy in the products. And because of that, we can use that energy to make ATP. So because the energy in the reactants is higher than the products, then the delta G is negative, and this is a spontaneous reaction. It doesn't require us to input any energy to do this. We might have to put a little bit of energy in it. If you think about like a rock sitting on a hill, right? You may have to put a little energy into that rock to get it to start moving, but once it starts moving, you can just stand back and watch the destruction, because the rock is naturally going to release its potential energy into kinetic energy, and it's going to roll downhill. So we don't require energy to do this reaction, just a small input of energy. And this is the energy of activation, which we're going to talk about in the next couple slides. So if we could do the math on this, 1 mole of glucose is 686 kilocalories to do work in the cell. So if you were to weigh out a mole of this, it would have 686. Remember, our nutritional label really says calories, but what it means is kilocalories. So a mole of glucose has 686 calories in it, and we can use that to do work in the cell by making ATP. And then that means the products have 686 kilocalories less energy than the reactants. One thing I want to point out here is that if we look at this equation, there's no difference between the reactants and the products. All the molecules are the same. There's 6 carbons on both sides of this reaction. There are there are 18. We got 6 plus 12. So there's 18 oxygens and 12 hydrogens. And so we can see this 6 carbons, that's 12 plus 6, that's 18, and then 12 hydrogen. So the molecules are exactly the same. The only difference is the way that they're structured, and what determines the way that they're put together, the way they're built, are the chemical bonds. So this is the only place we get the energy from, is the way that the molecules are shaped. Does that make sense?
Yeah.
So we have exergonic, and so again we have this word gonic, which means energy. Endergonic means inside. So this requires energy, absorbs energy, requires energy from its surroundings to make the reaction occur. So in this case, the reactants have less energy than the products. And you can think about it as a rock going uphill. So if we want the rock to go uphill, what do we have to do? We have to input energy into the system, you know, so you could push it up the hill, or you could carry it up the hill, or you could use a machine to make it go up the hill. But regardless, you're going to have to input energy to get this to go. And because you have to put energy into it, it's not spontaneous. Rocks don't normally run uphill. Water doesn't normally run uphill. So this is a non-spontaneous reaction. Any questions about this? All right, so on the test, I might draw something like this and ask you, is the delta G positive or negative? So all you have to do to answer that question is see which one has more energy. So this bar graph is lower than this. The reactants have more energy or less energy than the product, so this has to be a positive delta G. And I could do the same thing here. So the reactants have more energy than the products, and so this would be a negative delta G, and this would be a spontaneous reaction. Okay, so an example of this in a biological system would be photosynthesis. So plants take in carbon dioxide and water, and they give off, or they produce glucose and oxygen. So these are high energy molecules, right? Remember, we use these to produce our ATP, and these are low energy molecules. And remember, first law of thermodynamics says we can't create or destroy energy. So this difference in energy between the reactants and products has to come from somewhere. Can you guess where the energy comes from for photosynthesis?
The sun.
Yeah, it comes from the sun. And so that way we follow the rules of thermodynamics. We can't create or destroy energy. We can only change forms. So we have to put energy into the system to get it to occur, so that these are the same. All right. And then again, so this delta G is positive, it's non-spontaneous. We have to input 686 kilocalories per mole to produce this glucose molecule, and that comes from sunlight. All right, so this slide is just to remind you that we're open systems. We covered this in chapter 1. If I put you in a box, you wouldn't be able to get the things that you need. So, you know, like sunlight, for example, would produce food because you can't photosynthesize. If I did it to a plant, it wouldn't be able to produce any food. if I did it to you, you wouldn't be able to get any food, so you would die. You wouldn't be able to get any water, so you would die. So we have to get energy somewhere, and our energy system generally is from the sun, although there are other forms of energy, and we'll talk about that in a second. So if we put a reaction or a living system in a closed system, eventually it would equilibrium. That means that the rate of the forward reaction would equal the rate of the reverse reaction. And so you think about it like the other graphs that we were looking at. If there's no difference, then, and we have this rock, there's no energy being input or given off. What happens to that rock? It's just gonna
Just sit there.
Yeah. And so there's absolutely no energy available in this at all. And so if we get delta G is 0, right, the reactants and products are the same, then life couldn't exist, because there's no way that we can have an energy exchange. So the artist in your book did a drawing. Basically, they closed the system off. You know, with this, the example they're using is kind of like a hydroelectric dam. So we have you know, high energy on one side with the water, and then we use gravity to spin this turbine and make electricity, and that goes to a low thing. But imagine if the dam's level was the same as the other side, the water wouldn't move, delta G would be 0, and you would get 0 energy. And so without energy, we defined in chapter 1 that energy is required for life. So this equilibrium in metabolism is one of the features of living systems. And in order to fix that, all we have to do is get some external input of energy, like the artist is just showing a open spigot, which fills this side up and continues to allow this to generate electricity. You know, this is like rain, and water vaporizes into the air because of the sun, because the molecules move faster, they condense as they get colder and then fall back to earth. So this is kind of how the hydroelectric dams work, like the Hoover Dam. Okay, so that brings us to our energy molecule. Our energy molecule is ATP, which stands for adenosine triphosphate. Adenosine, this is the same molecule as we have in DNA and RNA. This particular, so it's a nucleotide, and we covered this in chapter 5. This particular nucleotide is ribose and not deoxyribose. So that's the A in RNA. Remember, RNA has A's and G's and C's and U's because there's no T's. And so this is the letter in RNA that we're talking about for ATP. And if we think about the molecule itself, here's our sugar. It has hydroxyl groups on the number 2 and the number 3 carbon. And then it has those 3 phosphates attached to it. And then it has this base. And you guys learned in chapter 5 that base here is an A. And if you forgot about what it looks like, you can go back to chapter 5 and check it out. But this is ATP, right? It's a RNA nucleotide. Because it has these 3 phosphates, and if you remember from chapter 4, what's the charge on those on phosphates? Do you remember?
Is it positive?
It's the other one.
Negative.
Yeah. So we have 3 phosphates with negative charges sitting next to each other. And so if you put 3 magnets with a negative end next to each other, what would they want to do?
Push away.
Yeah, they want to push away. So the thing holding these together are chemical bonds. And so imagine, we talked about the stick. Imagine if you were pulling on a stick really hard, you would weaken it. And so this is already inherently weak, and we just need something to give it a little extra oomph to break this bond. So ATP is kind of like a spring that you push down. So it's a compressed spring. And when we get rid of that phosphate, there's not as much pressure on this molecule. So it's more like a spring that's been, I guess, sprung. And so this has energy in it. You know, if you push down a spring and put a ball on it and let it go, it would shoot up. But if you didn't push it down and you just set the ball on it, it would just sit there. So this is, I don't want to say no energy, but it's really low energy. It's not really usable energy. So this is a high energy molecule. This is a low energy molecule, and this is really what we use to do work. And we already looked at the sodium potassium pump as an example of that, how we can add a phosphate to that integral membrane protein and cause it to change its shape so we can build that electric charge across the membrane. Okay, so ATP is a source of energy, mechanical. So if we add a molecule to another molecule, we change its shape. And we talked about this, that for you guys to be able to walk, you have to be able to change the shape of your leg and your body. So it's the same thing on a smaller scale inside your muscles. Transport, so we talked about the sodium potassium pump. We can use a change of shape to move things across a membrane. We also talked about the proton pump. So that's another example. And then we can use it to do chemical reactions. And we're going to cover that in 1 second, but this, by adding a phosphate, we can change the delta G of a molecule, right? So remember the bonds, and we talked about this. So if we have a bond, in order to break that bond, just like a stick, we need to bend that thing. In order to bend it, we have to change its shape. And that lowers the delta H, which lowers the delta G. So we can use ATP to change the shape of a molecule to cause a non-spontaneous reaction to become spontaneous. All right, so I kind of drew this out. I guess I didn't really have to. So here's the base, and we talked about the sugar ribose here, and remember that we number the carbons 1, 2, 3, 4, and 5. So in DNA, we wouldn't have this here, but it's RNA, so this is ATP. And then we talked about the 3 phosphate groups having negative charges and making it unstable. So just like every other reaction, if we want to break this bond, we have to add water, right? Hydrolysis, which we covered in chapter 5. And so we add water to that. We can remove the phosphate. We can add this to other things through other reactions, but we add water, we break that bond, and then this becomes ADP. So this is low energy, and this is high energy. And remember that first law of thermodynamics, we can't create or destroy energy. So if this is high and this is low, that energy has to go somewhere. And we usually transfer it to a molecule. And that changes its shape. So we can do work. All right. So the specific amount of this energy released is 7.3 kilocalories. You don't need to memorize that, but I'm going to go through sort of how this works. And so that's why I'm throwing this number out there. This is under standard conditions, and this is outside of the cell. So for whatever reason, inside the cell, it's closer to 13 kilocalories. So it's almost twice as much inside the cell as outside. Nobody knows why. So if you want a Nobel Prize, figure this out. I guarantee you'll win a Nobel Prize for that. It may take you a long time, but you'll get one. Okay, so phosphorylation is a term which simply means adding a phosphate. And so that's the whole key to this thing, is that we're going to take a phosphate off of ATP and add it to another molecule. The example your book uses is glutamic acid. So glutamic acid is a precursor to the amino acid glutamine. And if we took glutamic acid and we add ammonia to it, we end up with glutamine. But the problem is that this reaction's delta G is positive 3.4 kilocalories per mole. So is this spontaneous or non-spontaneous?
Non-spontaneous.
Yeah. And so this reaction would never occur on its own. We have to make this spontaneous. So that means we have to lower the delta G. And remember, we can't, in the body, we can only use delta H. We can't use T delta S to lower it, because if we increase the temperature or increase disorder, that would kill you. So this is all we have to work with inside the body without changing the temperature or disordering the system. So to do that, the body would take glutamic acid, which is right here. It would take ATP and add a phosphate to it. So remember, that phosphate has a negative 7.3 kilocalorie delta G. And so we can do simple math. Like, I don't expect you guys to do the math on this. This is what someone that studies chemical thermodynamics would do. But I'm giving you these numbers because I want you to see that we originally had a delta G of plus 3.4. But when we add that phosphate, we can add these values together. So 3.4 plus a negative 7.3 makes the net delta G negative 3.9. And so is this a spontaneous or non-spontaneous reaction?
Spontaneous.
Right. And so your body can turn a non-spontaneous reaction into a spontaneous reaction by reducing the delta G. And on a molecular level, it's the bonds, right? Everything has to do with the bonds. So really, that phosphate is causing that molecule to bend. And just like a stick, it weakens that bond. And that reduces delta H, which also reduces delta G. So that's how this works. It's the same way that you would break a stick. Then we have this phosphorylated glutamic acid. Now it combines with ammonium, and we get the reaction. And now, once the reaction's occurred, we can release this. This I means inorganic phosphate, which means it's not attached to a carbon anymore. So this is, we can make it an endergonic. So it's an endergonic reaction because we got to put energy into it, right? So we can make this a spontaneous reaction by adding that phosphate from ATP. Do you have any questions about that?
No.
All right, so we're going to learn in chapter 9 that the whole reason that you breathe oxygen is to make ATP. So we take the food that we eat, we combine it with oxygen, and the whole purpose of that whole thing is to make ATP. You use a lot of ATP. And just to kind of get an idea of how much ATP your body needs, you could hold your breath, and then when you held your breath, you would stop making ATP. So eventually you would run out of ATP. And once you did that, you would die. So your body uses massive amounts of ATP throughout the day. In fact, if you were just to look at a single muscle cell, the entire amount of ATP in that muscle cell is depleted once every a minute. So that's 10 million ATPs per second per cell for you to survive. And if we did an equivalency, that would be about 150 pounds of ATP for an average human per day. So do you think you could eat 150 pounds of ATP a day?
No.
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 So the only way to really do this is to recycle it. And so your body is really good at this. So remember, if we use ATP, it becomes ADP, high energy molecule to low energy molecule. But remember, all reactions are reversible. So what your body does is, instead of making a whole ATP from scratch, all it does is add a phosphate, right? And once we add the phosphate, we get ATP. So this is the key to regenerating ATP. This is endergonic, so it requires energy, and that energy comes from those high energy molecules like glucose and oxygen. Yeah, so lots of energy your body consumes. Any questions about what we talked about so far?
Nope.
Okay, cool. So that leads us to enzymes. So enzymes are proteins. I have lots of students that are taking advanced classes that never really got this. Remember, proteins are made of amino acids. And enzymes are workers. So you can think of them as workers in a factory. Their job is to do things to make reactions occur, because that's really what we are. Or to move things around or whatever. But they're simply workers. So think about it as like an Amazon factory. So if you ordered something, you wouldn't get the worker delivered in your box, right? The worker does its work. They pack the stuff, they ship it off, and then they can go do another job. So they can pack something else, and they keep doing that until they get exhausted, and then they get mad at Amazon and quit, or they go home for the day or whatever, and they rest. So enzymes, they can do their job many times, but eventually they wear out, just like everything else, which goes with that second law of thermodynamics, which is every reaction tends towards disorder. So these things are going to eventually break down, just like a car, just like the pyramids. So their job is to, so let's say that you ordered a phone case from Amazon. So, Amazon could have a factory where they just had a robot that threw phone cases around the room, and eventually maybe a phone case would fly in a box that had your address on it. And that could happen, but it would take a lot longer than having a worker go and pick the phone case and stick it in a box and put your mailing address on it. So the reactions we're talking about would occur on their own eventually, just by random molecules bouncing around. But what enzymes do is they direct the reaction. And because they do that, they can speed up the reaction. And anything that speeds up a reaction is called a catalyst. So a catalyst accelerates a reaction. But because not all catalysts are proteins, like there's a catalytic converter in your car that reduces pollution emissions, and that's made out of metal. So an enzyme is specifically a biological catalyst, and it's made out of proteins, and its job is to speed up a reaction. So this is defining catalyst, which I just talked about. But remember, a catalyst speeds up the reaction. It doesn't become part of the reaction. It's not packing itself in your box. If your job was to put tires on a car, you wouldn't become part of the car. You just put the tire on and then go over and put another tire on another axle, and so on and so forth, until you got tired or quit or whatever. So to summarize, enzymes are catalysts, which means they speed up reactions. They're made of proteins, which are made of amino acids, which makes them biological. They speed up reactions that would naturally occur on their own, but they go faster by directing the reaction. And they don't become part of the reaction, and they're not used up in the reaction. So they can do this over and over again, which is kind of the definition of a worker. All right, so the reaction might occur on its own, but it would occur really slowly. You know, think about if your job was putting tires on a car and you sat in the corner and threw it at the car. So eventually it might bounce on there and land on the axle, but chances are that's going to be few and far between. So we want to direct this. And because of that, enzymes are really important, because you know that you have to you have to react quickly to your environment in order to be able to survive. So to summarize, enzymes speed up reactions. Enzymes always end in ASE. So here's an example of an enzyme, DNA polymerase. This is the enzyme that copies your DNA to pass on to new, what we call daughter cells. Okay, so a lot of students have a problem with this, and I kind of get it, but I'm going to try to explain this the best that I can so that you guys can understand what enzymes do. So this drawing shows the reactants. So, you know, they're just using A and B, but it could be oxygen and glucose, right? It's just showing these bonds. Remember, all the energy is in the bonds. And so in order to, you know, if we're taking glucose and oxygen, 6, and we're making carbon dioxide, it's also 6, and water, it would be like we have all the parts, right? The parts are the same, and we just went over that. So in order to turn this into this, we have to take the parts apart. In other words, we have to break those bonds. If you can imagine, it's like a chemical modeling set, and I gave you a glucose and an oxygen and said make carbon dioxide and water. You'd have to take the balls apart from the sticks and rearrange it in a new way. So it's the same thing here. There's a transition state where the bonds are weakened, right? And that's what the squiggly line is. And what enzymes do is cause this transition state. So remember, we have our bond, let's say it's between oxygen. So there's 2 oxygens. And in order to break that bond, just like the stick, we have to bend it. So we add a phosphate to that bond, it bends the stick, and that's what this squiggly line represents. So the enzymes aren't part of the reaction. What the enzymes do is they're responsible for adding the phosphate to the molecule to weaken the bond. So the enzyme would add the phosphate here to weaken these bonds, and there is a little bit of energy that has to be put into it to add the phosphate to it. And that energy comes from the ATP molecule, right? So this energy of activation is what enzymes do, or enzymes overcome to get the reaction to start. So let's go back to our rock example. We have our reactants and our products. Reactants, products. And so we talked about that the rock rolls downhill, right? But we also talked about the rock's going to sit there unless we put some energy into it. So to get it to start moving, you might have to push it a little bit. So you have to put a little energy into it. that little bit of energy to start this going, to start the rock moving, to get these bonds to break, is called activation energy. And this is what enzymes can change. They lower this activation energy. And so it's easier to get the rock to start going. It's easier to start to make that non-spontaneous reaction to become spontaneous. And in the end, our goal is to just rearrange the bond. So here you can see that the A was bonded to B and now it's bonded to C. And B was bonded, or C was bonded to D, and now B is bonded to D. So this is the products. The delta G here is going to be negative because the energy of the reactants is higher than the energy of the products. The difference of that energy is this change in delta G, which would be negative. So this is a spontaneous reaction, but we still have to use the enzyme to input the activation energy to get it to start the reaction. All right. So transition state is when we weaken the bonds, and then the reaction will go until the bonds reform in a new way to make a new molecule, just like converting sugar and oxygen into carbon dioxide and water. All right. So I'm just going to tell you straight up, you're going to see something like this on the test, but this won't be on here. And I'm going to ask you, what does this graph represent? And because it's higher, so we already know this is the energy of the reactants. This is the level of the reactants. This is the level of the products. And so the difference between those, this arrow right here represents delta G. So because the reactants are higher than the products, is this the delta G negative or positive?
Negative.
Right. So rocks roll downhill, right? So you're right. It is not positive, it's negative. And I might ask you what this big line represents. So that is the reaction without an enzyme, because this hill, this activation energy is higher than this one, this red one, which would be the reaction with an enzyme. Oh, this is terrible. Reaction with an enzyme, because this is lower. And I might ask you, what's this arrow represent? Well, that's the difference. That's the activation energy, the EA, energy of activation, with the enzyme. And what does this arrow represent? This is the EA without the enzyme. So guaranteed this will be on the test. So I'm giving you a free study guide right here. Remember, enzymes do not change delta G, right? With or without the energy enzyme, the delta G is not different, right? This is kind of confusing, but remember, it's not the enzyme that changes the delta G. It's adding that phosphate, which can change the delta G. And remember, enzymes are specific because they have a specific shape, and that shape dictates their function. Okay, so enzymes are specific for substrate is what something the enzyme reacts with. So if you're thinking about an enzyme as a worker, and a substrate might be a car tire. So the enzyme responsible for working on cars, right? Maybe another enzyme is responsible for working on boats, and maybe another enzyme is responsible for putting the screen on a cell phone or whatever. So you can think of it as an assembly line, And these are all different. So like, for example, sucrose has a specific shape. And the enzyme that works on sucrose is called sucrase. Nothing else can work on sucrose to break it into, this is a disaccharide. And we talked about this in chapter 5. And these are monosaccharides. So sucrase adds water, right, to do that hydrolysis reaction that breaks sucrose into glucose and fructose, which is what it's made out of. And then there's, so lactose is the same. It's a disaccharide. It's galactose, which is a weird sugar, and glucose together. And so if you you want to take this disaccharide and turn it into monosaccharides, then you need an enzyme. What do you think the enzyme's called that breaks down lactose?
Glucose.
Remember that all enzymes end in ASC, so sucrose is,
Lactase.
yeah. And remember we talked about a sugar called amylose. So what do you think the enzyme that breaks that down is called?
Amylase.
Awesome. Yeah. So generally the enzymes describe the molecule they're working on. So the substrate here would be lactose or amylose or sucrose, and the enzyme would be sucrase or amylase or lactase. And so lactase doesn't work on sucrose or amylose. Amylase doesn't work on sucrose or lactose. So they're all specific for what they work on. And you know, we talked a little bit about this because we have these amino acids that are linked together. And we talked about their R groups. We talked about this in chapter 5 lecture. But we can make these R groups. So in the case of sucrase, its R groups are want to attract water, right? So we want this reaction. We need water to make this reaction occur. So sucrase would have amino acids that are hydrophilic because it wants to bring water in to break the chemical bond in sucrose so that it makes it releases it as glucose and fructose. You know, and if we wanted to reverse this reaction, then we would need an enzyme that was that had R groups that were hydrophobic because we want to scare water out, right? Because we need to remove water. I'm sorry. Yeah, we would need to remove the water in order to reverse this reaction. So and that would be a dehydration reaction. Okay. So the area where the reaction occurs, enzymes are big. Like this molecule here is glucose. So glucose is C6H12O6. It's not a small molecule by any means. It's made up of, you know, 24 atoms. So, you know, in the molecular world, that's relatively big. And it's shown here in red. This is the enzyme that reacts with glucose. And this molecule is called a hexokinase. We're going to see this again in chapter 9. But the first step, so you would eat amylose, right? This is the same word as carbs. In your mouth, you have an enzyme called amylase. And remember, amylose is glucose. This is horribly hard to draw a hexagon on this drawing pad. Anyway, So remember glucose is a hexose. This is my attempt at drawing hexagons. And they're linked together by those glycosidic bonds that we learned about in chapter 5. So amylose is long chains of glucose, and in order to digest that, to release the energy, you have to break it down. Well, that breakdown starts in your mouth with this enzyme amylase, and amylase fits over amylase has a specific shape, and it goes across two glucose molecules. So this is the amylose. Sorry, amylase. Amylase is here. This one is amylose. And so it breaks this bond right here, that glycosidic bond that we talked about in chapter 5. And so when it does that, it creates maltose, which we also talked about in chapter 5. So two glucoses linked together is a maltose. So that's a disaccharide. Now the digestion isn't done because we have to break down the disaccharide into a monosaccharide. That monosaccharide is glucose because maltose is two glucoses linked together. So in your stomach, that maltose gets broken into glucose. And we talked about that glucose, after it leaves your stomach, goes to your small intestines. And in your small intestines, the sugar level is higher than in your blood. So that goes from your small intestines to your blood. And then it travels around to your cells that need it. So the cells, before the cells open up channels for glucose to come in, they require insulin. Insulin gives a signal to produce a channel, that integral membrane channel, to allow glucose to pass across the membrane, because glucose isn't small, and it's certainly not hydrophilic, phobic, sorry. So it's not going to be able to pass through that membrane without a channel. And insulin sends a signal to build that channel. So once that occurs, then it goes into your cells. And so this is inside a cell. This hexokinase is just hanging out in the cytoplasm. And glucose binds to hexokinase. And this word kinase, and I want you guys to know this. This means it's an enzyme, right? So ASE means it's an enzyme, and its enzyme is to add a phosphate. It phosphorylates a molecule. So glucose comes in to your cell. It encounters this hexokinase. The glucose goes into the active site. It fits in here. When we add a molecule to another molecule, what does it do to it?
Changes the shape.
And so you can see this thing, it's sort of changed its shape to kind of enclose this glucose molecule. And that's where it reacts to add a phosphate. And so I'm going to download this real quick. So I'm gonna download chapter 9 so I can just show you what this looks like, because we're gonna cover this next. Okay, so Okay, so this is chapter 9, and I'm gonna scroll ahead just to show you what's going on. So so this is a process called glycolysis. And then just stop me if you can't see this slide from chapter 9, but should be energy investing phase.
Yeah, it's on it.
Okay, so this is what happens. So glucose comes into your cell. You have cell membrane. There's a channel that's built by insulin. Signals the DNA to build this channel. Glucose comes into the cytoplasm, and that's where we're at. So this is all occurring in the cytoplasm. And this process is called glyco, which means sugar, and lysis. Remember, the word is to break apart, like hydrolysis, using water to break apart molecules. So glycolysis is breaking glucose apart. So this is that hexokinase that I was just showing you. And its job of all kinases is to add a phosphate. That phosphate comes from ATP. So we make ADP. And then you can see that there's a phosphate that's been added to this glucose. So it becomes glucose 6-phosphate, because if we numbered this from the oxygen, this would be 1, 2, 3, 4, 5 right here. And then this is the 6 carbon. So glucose is, this is a glucose molecule with a phosphate added to its number 6 carbon. That's what hexokinase does. Now, I'm just going to jump ahead just to show you that. Remember, phosphates have negative charges. And so we're going to cover this again, but we want to add these phosphates on here. And we have negative charges on both sides of this molecule. What do you think it's going to do to this molecule?
Push it apart.
Yeah, so it splits it right down the middle. And remember, when we break down molecules, we're digesting them, and that releases energy. So that's the whole purpose of this hexokinase. It's the first phosphate load in this process to split glucose in half. All right, so I'm gonna go back to chapter 8, and this is what's going on here. So this area right here, which is a very small part of this molecule, this enzyme, the area where the reaction occurs right here is called the active site. So the rest of this enzyme might do other things. The important part for the reaction is this area right here where it binds, and it adds that phosphate. So that's what we mean by active site. It's usually a pocket or a groove on the surface of an enzyme that fits the substrate. So remember, the enzyme here is the hexokinase, and the substrate is glucose. Make sure that you guys know these terms, especially substrate, because it's not a normal term. It's just used in biology and science. Remember, it's only made by a few amino acids on the protein, and in this case, you know, we would want to transfer a phosphate. The shape is the function. It's kind of a lock fitting a key. So if we change this active site, it won't fit anymore. And I can tell you guys, there's a disease called Snyder-Robinson syndrome. One of my students had 2 boys that have this. It's a super rare, we call it an orphan disease, because there's only about 12 families on Earth that actually have this. And so we were trying to figure out what mistake was going on in this enzyme. It's called spermine synthase, and it has to do with these organic molecules. I don't want to get too much into it, but we discovered that the mutation that they had caused this active site to change its shape, And so our target was to see if we could figure out some way to change it back so that the molecules would fit again. And I would say that 99% of human disease is caused by some mutation that changes the shape of an enzyme that causes it not to function anymore. So this is a really important concept, especially if you're going into medicine. We call it, go ahead.
Is it hard to figure out how to change those little pockets back?
Yeah. So we worked with the University of Arizona pharmacy department, looking at different compounds that in the, in a test tube. We had their skin cells and we grew them in a test tube, and then we experimented on those. And so we went through about 4,000 different compounds to try to get something that would bind there to change it to the right shape, and we couldn't do that. So in the end, we ended up using a technique that's pretty prevalent now, and that is called CRISPR Cas9. You can read about it. It's beyond the scope of this class, but it's a bacteria viral defense, and you can use that to edit genes. So we went in and edited those genes to repair the mutation back to the way it was before. We still haven't figured out how to get it past the blood-brain barrier, but that's sort of the work that you might need to do if, like sickle cell anemia is caused by change of an active site, change of a shape of a protein. Like Huntington's disease, Lou Gehrig's disease, ALS, Snyder-Robinson, like tons of diseases are caused by mutations that change the active site of enzymes so they no longer function. And so lots of research dollars are going into how to edit these genes back to normal.
Is that kind of the same thing with the cancer cells you're talking about? How they start pulling the blood?
Yeah,
Do they
so
change it with that?
right. So yeah, so a lot of cancer biologists. So we're going to cover this in chapter 12 and 13, but there's a we're going to go over the cell cycle in chapter 12, but I'm just going to briefly do it. So we have a G1 and then a G2 and an M phase, and the whole purpose of this cell cycle is to make two new cells. We call them daughter cells. And in order to make good cells, we have to make sure that the DNA, so this is synthesis, and this is where DNA is copied. So I think I talked about this briefly, but if you imagine that you went out in the sun and you got sunburned, that actually causes damage to your DNA. The ultraviolet light makes two T's in DNA that are next to each other, covalently bonded, and those are strong bonds. So the DNA actually has to go in and repair that. Enzymes cut it out, and then they put new T's in. So every time you're out in the sun, when UV light hits if it gets into your nucleus and it hits your DNA, it will cause those T's to become stuck together, and your body has to fix that. So the longer you're in the sun, you can imagine the more damage is done and the more repairs have to be made. So let's just say that you have 10 workers in your cell. And so maybe one worker could fix a damage every minute. But what if you have 1,000 damages a minute? Then those workers are going to get overwhelmed. And eventually they're going to be like, I can't fix this. It's too much. And when it does that, there's an enzyme that checks the DNA. It's called P53. And P53, if the damage can't be fixed, it tells the cell to do apoptosis, which we talked about in the previous chapter, at chapter 6, where the lysosome opens in the cell, and this is cell suicide. And this is what causes sunburn. So your worker cells, they give up, they can't fix it. P53 sends it down this path to kill itself. In a lot of cancers, this is mutated, so it's broken and it doesn't work. So the enzyme can't check the cells, and so instead of going to cell suicide, they get passed on with all those mutations. And you guys probably know that the more mutation you accumulate, the closer you are to cancer. And so 99.9% of cancers are missing this enzyme. So a lot of clinical trials are being done to reintroduce or fix this enzyme. You can kind of imagine it as a bouncer in a club. So, the bouncer's there to only let certain people in and to exclude other people. Maybe you're not dressed nice or whatever. So imagine the bouncer leaves. It's got to go to the bathroom or something. So then everybody gets in, right? Whatever riffraff is in there. And that's kind of like what P53 does. So it doesn't let messed up cells go through the cell cycle. It sends them to kill themselves. But if it's gone, there's nobody checking. And so everything gets passed on, which makes more and more mutations, which leads to cancer. Now imagine that if you could get the bouncer to come back, then it would be like, whoa, wait, how did you guys get through here? You get over here in the kill yourself line, and you too. And so by reintroducing P53, the idea is that it will detect these bad mutations when this cell goes through this next cell cycle. P53 is there, and it's like, wait, you're bad. You go kill yourself. And that way you can get rid of cancer, because all the cancer cells would be instructed to kill themselves. So that's an example of an enzyme that we could mutate to maybe cure cancer. And trust me, there's millions of dollars being poured into figuring out how to fix this P53 enzyme.
Cool, thanks. That's really interesting.
Yeah, no problem. Yeah, I mean, we spent almost 10 years trying to figure that out in breast cancer, and now what they're doing is they're kind of like using it like the Pfizer vaccine. So they just make the P53 mRNA, and they put it in a lipid vector, and then that gets delivered to the cells, and the cells make P53 from that mRNA. It's the same way that the vaccine works, and that's really the research on like P53 and stuff is how we had the technology to be able to build that vaccine so fast. But that's what they're doing with clinical trials right now, and it appears to be relatively successful. So, high hopes. Anyway, so this is how an enzymatic reaction works. Here's our substrate. In this case, it's sucrose, but it can be anything. It could be glucose, or it could be whatever. So sucrose is a disaccharide. It's made up of fructose and glucose. You can see that here. And it's linked together at that glycosidic bond. So the enzyme, the substrate binds to the enzyme at its active site. Make sure you know that name. Active site. And remember, the enzyme is going to be named usually after the substrate. So in this case, it's sucrase. It binds. Remember, we have to add water to break the bond. And that's a dehydration reaction that we learned in chapter 5. And then once that bond is broken, we release fructose and glucose individually. And then again, glucose would go to that hexokinase, and it would eventually get broken down further to make ATP. Same thing with fructose. It would end up, it's not the same molecule because this is more shaped like a pentose. So there's another enzyme called a pentokinase that would react on fructose, and that would send it on its pathway to split it in half to make ATP as well. And then once it's released, the enzyme can react with a new sucrose. So if the enzyme isn't used up, it just keeps going. Eventually, though, that the second law of thermodynamics will make this sucker wear out, and it'll have its right shape anymore, and then you have to make a new enzyme. So that it doesn't last forever.
Does it just get broken down once it's used up?
Yeah, so once it doesn't function anymore, there's a whole system. If you guys took a cell biology class, you would learn about this, but there's a whole system to destroy proteins, and then they get recycled. So the amino acids just get split apart and then rearranged into new proteins. So this could end up being, you know, insulin or something like that. It could be it could be hemoglobin. It's only the order of the amino acids makes one protein different from another. Kind of like what we covered in chapter 5. All right, so I kind of went over this, but I'll just kind of blast through that, and then I'll end the lecture here. So enzymatic reactions, remember the active site holds the substrates. These are the things that it's reacting on, enzymes react on. It could be weak interactions, like we talked about the weak reactions. Hydrogen bonds and ionic bonds, remember, we're inside the cell, so this is, ionic bonds are weak in the presence of water. The active site can distort the substrate. Chemical bonds, like we're talking about bending it, right? So it lowers the energy, that delta H energy. It could make a small environment where, like we talked about, it might be hydrophobic or hydrophilic. Maybe it changes the pH a little bit, and that causes the reaction to occur. So there's all kinds of things in those R groups. Remember, they could be positive or negative. They could be acidic or basic. They could be hydrophobic, or hydrophilic. So any of those microenvironments that active site would make. So this is the active site. Could cause the reaction to occur where it wouldn't normally occur. Well, it would, but it could take a year or 1,000 years. So the enzyme's speeding that reaction up. So the rate of the reaction depends on the concentration of the substrate. That makes sense. If you're building iPhones, for example, if you don't have enough parts, then you're only going to be able to make so many iPhones. You want to make more iPhones, you need more parts. So you're not going to increase the number of iPhones or the speed that iPhones are made if you don't increase the amount of parts. In this case, you're not going to make more fructose and glucose unless you increase the amount of sucrose. So the more substrate, the faster the reaction. But remember, every cell has a limited number of workers. It's just like a factory. So if you only had 10 people working in your iPhone factory, maybe each person can make 10 phones a day. You're only going to get 100 phones. So we call that enzyme saturation. That means that these enzymes can only do so much work. So if you wanted to make more iPhones and you had lots of substrate, how could you make more iPhones?
Add more enzymes?
Yeah, you just hire more workers, right? You add more enzymes. And that overcomes that saturation. So, I mean, I'm giving these examples because I want you to realize that everything that's going on in this little molecular tiny world is the same sort of junk that happens in real life, in factories and stuff like that. It's just on a smaller scale, and I try to want to make it relatable so that you guys can see that this isn't like magic. It's the same thing that you experience every day. You just got to think of it in like a macro environment instead of a micro environment. But everything is basically the same inside your cell, except your workers are called enzymes and they're made out of amino acids. But they all have jobs to do, and you know, it's not magic that your muscles move. It's adding phosphates to change their shape. You know, coordinating that is a pretty crazy feat, but the small level things that it does is pretty standard, you know, as far as looking at our actual world that we interact with every day. All right, so I'm going to end here, and then hopefully I'll remember to post this onto YouTube. If you have any questions, I won't be here Friday. I have a department meeting, so and then we have spring break next week. So we'll pick this up on, let's see. I'll have office hours on the 22nd, and then the next lecture will be on the 23rd. I'm not sure when your test is coming up. Is it the 31st?
I think so.
I might need to
Yeah.
move that test date a little bit because I'm not sure we're going to get through all the material, but I'll know more after we come back after spring break. So office hours 22nd, and then next lecture will be the 23rd. So you guys have a good spring break, and I'll see you in a week and a half.
Thank you.
All right, have a good one.